I had a moment this morning in traffic, actually.
Yeah.
Yeah, and it really sort of it set the stage perfectly for what we're about to dive into today.
Okay.
Something cut me off. I mean, it wasn't even that bad, just a standard, everyday lane change.
The usual.
The usual, exactly.
Yeah.
But for maybe, what, 3 seconds?
Yeah.
I wasn't myself. I was just this ball of pure, unadulterated fury.
Ah, the classic road rage reflex.
Exactly. But here's the thing that gets me. I think of myself as a rational person.
Mm-hmm.
I pay taxes, I read books, I get how the physics works. My logical brain is fully aware that getting angry at a Honda Civic doesn't change anything. It solves nothing.
Of course, the logic is sound.
But in that moment, the logic it wasn't there. It didn't matter. Something else was at the wheel.
You were hijacked.
I was completely hijacked. And that is exactly what we're getting into today. We spend so much of our time thinking of our brains as these, high-powered logical supercomputers.
It's a very clean metaphor, yeah. It's super clean. You input data, you run a calculation, you get a decision. It feels very orderly.
It's a comforting idea. It makes us feel like we're in control,
Yeah.
that we're rational agents acting on pure logic.
But then you get those moments. The road rage, or suddenly being so hungry you can't think, or that wave of anxiety that comes from nowhere. And you realize there's this whole other layer, a deeper, older, almost grimy layer, that's actually deciding why we do anything at all.
That is the perfect way to frame this. That's the deep dive for today. We are looking at the literal hardware behind your feelings, your drives, your wakefulness, even your memories.
We're talking about the limbic system and the hypothalamus.
Precisely.
And just to set expectations for you listening, we're going to get incredibly specific. We're going line by line through chapter 59 of our source material here.
Yeah, this isn't a high-level overview. No. We are just going to say the brain controls emotion. We want to look at the specific biological machinery, the actual nuts and bolts that makes you feel awake or hungry or angry or happy, and what allows you to learn anything in the first place.
The official term for this is the behavioral and motivational mechanisms of the brain. And I think before we get into the really complex wiring, we should start with that word limbic.
Oh, yeah.
Because it gets thrown around constantly in pop psychology. Oh, that's just my limbic system acting up.
Right, it's my lizard brain. That's the one I hear all the time. But what does limbic actually mean? Where does it come from?
Well, etymologically, it's from the Latin word limbus.
Limbus.
Okay, which means border.
A border,
Yeah.
like a boundary line on a map.
Exactly that. Originally, anatomists used the term to describe the border structures that were wrapped around the basal regions of the cerebrum.
So it was just a geographical term.
It was purely geographical. They looked at a brain and saw this ring of structures separating the deep central part from the big outer cortex, and they just called it the limbic lobe, the border lobe.
So when they first named it, they didn't even know what it did. It was just, here be the borderlands.
Precisely. It was a location on a map. But of course, over time, as we started to understand the function of these areas, the definition totally shifted.
Right.
So today, when a neuroscientist says limbic system, they're not just talking about a border, they mean the entire neuronal circuitry that controls our emotional behavior and our motivational drives.
It went from being a place to being a functional system.
Yeah.
An interconnected network.
Yes. And if you look at the diagrams of that network, right in the center of that whole web, you find one key player.
The hypothalamus.
The hypothalamus. It's absolutely the key. It's physically at the center, and it's surrounded by all these other subcortical structures. The septum, the amygdala, the hippocampus, the cingulate gyrus.
All the famous names.
All the big ones. But functionally, the hypothalamus is the headquarters. It's the CEO of the entire limbic system.
And we are definitely going to get deep into the hypothalamus later because the list of things that tiny piece of brain does is just insane.
It's staggering, yeah.
It's regulating everything from your body temperature to your sex drive. But first, I want to step back a bit because before we can have emotions, before we can be hungry or angry, well, we have to be awake.
Correct. You have to turn the machine on.
And the source material brings up this idea that I found genuinely humbling. The concept of the useless cortex, which sounds a little harsh for the part of the brain I'm using to host this deep dive right now.
It is harsh, but it's biologically spot on. We have this tendency to think of the cerebrum, that's the big, wrinkled outer part of the brain where consciousness seems to live. We think of it as self-sustaining.
Like a laptop that just runs on its own battery.
Right, but it's not. It's not a laptop. The truth is, without continuous nerve signals coming up from the lower brain, the cerebrum is effectively useless.
So it's more like a like a hard drive. You can have all the data in the world on it, all your memories, your personality, your ability to do calculus.
Yeah.
But if you don't plug it into a power source, it's just a paperweight.
That's a perfect analogy. And we see the tragic reality of this in medicine. If there's a massive compression of the brainstem, specifically at the juncture between the mesencephalon and the cerebrum, the person goes into an unremitting coma.
And that can happen even if the cortex itself is perfectly healthy, no damage at all.
Yes, and that's the terrifying part. The hard drive is intact. The person, structurally, is all there.
Wow.
But the power switch has been flipped off. And that power switch is what we call the reticular excitatory area.
Okay, let's map this out. If this is the thing that's keeping me conscious right now, I really want to know where it is.
It's located deep in the brainstem, spanning the pons and the mesencephalon. It's also sometimes referred to as the bulboreticular facilitatory area.
Okay, so it facilitates, it makes things happen.
Yeah.
And its job is to send signals up to the cortex.
It's really a two-way driver. It sends signals down to the spinal cord, for one. That's what maintains the tone in your anti-gravity muscles and controls your reflexes. It's why you don't just collapse in a heap when you stand up.
Right.
But crucially for this discussion, it sends this profusion of signals up to the thalamus, and from there, out to the entire cortex.
And the source mentioned there are actually two different kinds of up signals. It's not just one big on button. It's more nuanced.
Exactly. You've got a fast system and a slow system. The first type involves really rapid action potentials.
Okay.
These signals come from large neuronal cell bodies in that reticular area, and they release a specific neurotransmitter.
Acetylcholine?
And these are the fast ones, you said.
Extremely fast.
Yeah.
They excite the cerebrum for only a few milliseconds. It's like a spark, a flash. It creates an immediate, sharp awareness.
So that's the spark. That's me suddenly noticing the traffic light just turned green.
Exactly. But that's not enough to keep you awake all day. For that, you need the second type of signal.
The slow one. The slow background excitation. These signals come from a huge number of tiny neurons. They travel up to the thalamus, but instead of just one big spark, they synapse with these small, slowly conducting fibers that then trickle out to all regions of the cerebral cortex.
So if the first one is a spark, this one is more like turning up a dimmer switch, maybe.
That is a great way to put it. Or like the background hum of an engine that's always on. The excitatory effect from this system builds up progressively over seconds or even minutes.
And this controls the long term. What does the source call it? Background excitability.
That's the term. It sets the baseline for how ready you are to process information. Are you groggy and slow, or are you alert and ready to go? That's the dimmer switch.
Now, this is the part that I found really, really interesting. It's not just a one-way street, is it? It's not just the brainstem yelling, "Wake up!" at the cortex. The cortex talks back.
It absolutely does. It's a positive feedback loop, and it is crucial for understanding why we stay awake.
So how does that work?
The cortex, when it gets activated by thought processes or motor processes or anything really, it sends signals back down to that same excitatory area in the brainstem.
Okay, let me see if I've got this straight. The brainstem sends a signal that wakes up the cortex.
Mm-hmm.
The cortex starts thinking about something. Maybe I'm worrying about a deadline for work.
Yep.
That act of thinking and worrying sends a signal back down to the brainstem, basically saying, hey, we're busy up here. Keep the power on.
Exactly. And that feedback makes the brainstem even more excited.
So it sends even more wake-up signals back to the cortex.
Which makes the cortex think even more, which excites the brainstem even more, and on it goes.
That explains so many sleepless nights.
It's a self-sustaining cycle of wakefulness.
The classic advice, just stop thinking, is technically the cure, but you can't. Because the thinking itself is what's keeping the power on. You're trapped in the loop.
Precisely.
What about external things? Like, why is it impossible to sleep when you're in pain?
Well, that's the other major input to this entire system. Sensory signals coming in from the body, especially pain signals, feed directly into that reticular excitatory area and give it a massive jolt.
So pain is like a shot of adrenaline straight to the brain's on switch.
A very powerful one. And the source gets really specific with the anatomy here, mentioning the fifth cranial nerve.
The trigeminal nerve.
Yes. This is the highest nerve that enters the brain, carrying a significant amount of sensory information, mostly from your face and head.
And there was a fascinating, if a bit grim, experiment that proved this point.
It's a classic in neuroscience. If you make a cut in the brainstem above the point where that fifth nerve enters, The animal falls into a permanent coma.
Because you've severed the connection from the power switch to the cortex. You've unplugged the computer.
Exactly. The sensory input can't get to the excitatory area anymore. But here's the kicker. If you make the cut below the fifth nerve,
The animal stays awake.
the coma is averted, yes.
Wait a minute. Even if you cut off all the sensory signals from the arms, the legs, the entire torso.
Even then, the sensory input from just the face and head coming through that one nerve is enough to keep the brain stem firing and the cortex awake.
Wow. That just shows how dependent our consciousness is on this constant stream of sensory data. We need to feel things just to be awake.
Our brains are not designed to exist in a vacuum.
Okay, so that's the gas pedal. The reticular excitatory area gets us going and keeps us alert. But you can't just have a gas pedal. You'd burn out in days. We need a break.
We absolutely do. And that brings us to the reticular inhibitory area.
Location check on that one.
It's located medially and ventrally in the medulla, so a bit lower down in the brainstem than the excitatory part.
And its job is just to, what, calm everything down.
Its job is to inhibit the excitatory area, to put the brakes on. And the mechanism here is crucial because we're moving from purely electrical signals to neurochemistry.
Right.
The inhibitory area works by exciting what are called serotonergic neurons.
Serotonergic, meaning they secrete serotonin.
Yes. And in this particular circuit, serotonin acts as a depressant. It depresses overall brain activity. This system is absolutely essential for sleep.
So if that serotonin system isn't working right,
then the excitatory area just runs unchecked and you get severe insomnia. You simply cannot sleep.
this is a perfect transition into what the source calls the neurohormonal systems. And I love this section because, we often think of the brain as just wires. Neuron A connects to neuron B, like a circuit board.
A very point-to-point direct model.
Exactly. But this part of the chapter describes it more like a chemical soup.
That's a great distinction. The electrical signals we just talked about are precise. They are fast. They carry specific data. I see a red car. I feel a sharp pain.
Right.
But these neurohormonal systems are broadcasters. They release chemicals that diffuse through the substance of the brain, and these chemicals can persist for minutes or even hours.
So they're not sending a specific message. They're providing long-term control.
They're setting the mood of the brain. It's the difference between sending a text message to one specific friend and pumping a gas into an entire room that changes everyone's mood simultaneously.
That's a perfect analogy. Okay, the source lists 4 main systems here. I want to go through them because they seem to control pretty much our entire state of mind. System number 1, the locus coeruleus.
A tiny, tiny area with a massive impact. The nerve fibers from the locus coeruleus spread out everywhere, the cortex, the cerebellum, down the spinal cord. Just an incredible distribution.
And what chemical is it broadcasting?
It secretes norepinephrine.
Norepinephrine. We usually think of that as an excitatory hormone, right? Like an adrenaline for the brain.
Generally, yes. In most areas, it excites the brain, leading to increased activity. But biology is always full of contradictions.
Of course.
In a few specific brain areas, it actually has inhibitory effects because of different types of receptors on the neurons there. But the big headline for the locus coeruleus, the most interesting function, is its role in REM sleep.
In dreaming.
Exactly. This system is a major player in generating the rapid eye movement sleep state where our most vivid dreams occur.
So wait, the same chemical that generally wakes you up and makes you more active is also responsible for the most active phase of sleep. That seems like a paradox.
It does, but think about what a dream is. Your brain is hyperactive, you're running, you're flying, you're having intense emotional experiences. Your brain is very much awake inside the dream.
Okay, so that's the norepinephrine at work. That makes sense.
Precisely. Okay, system number 2, the substantia nigra.
That sounds like a villain's secret lair, the black substance.
Well, it actually looks dark in unstained brain tissue, which is where it gets the name. It's located in the mesencephalon.
And its neurons send fibers up to the basal ganglia, specifically, the source says, the caudate nucleus and the putamen.
Correct, and they secrete dopamine.
Dopamine, the famous one. We always hear about it in the context of reward and pleasure and addiction. I got a dopamine hit from that social media like.
And that's true, but that's in other dopamine pathways. In this specific pathway, the one going to the basal ganglia, dopamine acts as an inhibitory transmitter.
It calms things down.
Yes, and the source notes a critical clinical correlation here. The destruction of these dopamine-secreting neurons in the substantia nigra is the direct cause of Parkinson's disease.
Right, because without that dopamine inhibition in the basal ganglia, the motor system goes haywire.
It becomes overexcited.
And you get the rigidity, the tremors, the loss of controlled movement, because that crucial calming signal is just gone.
Exactly. Now, system number 3, the Raphi nuclei.
These are found in the midline of the pons and the medulla.
And they secrete serotonin.
We just talked about serotonin a minute ago.
Right.
That's the brake pedal for the sleep system.
That's right. Fibers from these nuclei go up to the diencephalon and the cortex, and their release of serotonin helps cause normal sleep. But, and this is fascinating, they also send fibers down the spinal cord.
Oh, four.
To suppress pain.
Wait, really? The same chemical that helps you fall asleep also acts as a natural painkiller.
It does. It's a key part of the body's own analgesic system. And it makes perfect sense when you think about it. When you need to rest and recover, what 2 things do you want to do?
You want to shut down your conscious mind, and you want to turn down any pain signals.
And serotonin does both. It's incredibly efficient.
Okay, and the last one, system number 4, the gigantocellular neurons.
A mouthful, I know.
Yeah, these are located back in the reticular excitatory area where we started. They release acetylcholine. And as we already discussed, this leads directly to an acutely awake and excited nervous system.
So to recap, we've got this incredible chemical cocktail constantly swirling around our brains. Norepinephrine, dopamine, serotonin, acetylcholine, it's not just wires. It's a delicate, shifting balance of fluids.
And that balance sets the stage for everything else that happens. But now we need to move up the chain of command to the structure that actually manages the body's day-to-day survival while we're awake. The CEO of our vegetative functions.
The hypothalamus. The hypothalamus.
We mentioned it's the center of the limbic system, but I want to re-emphasize just how physically small this thing is.
It weighs about 4 grams.
4 grams. That's nothing.
It's less than 1% of the total brain mass. It's minuscule.
And yet, the source makes a very bold claim. It says if you damage your cerebral cortex, you might lose a function. Maybe you lose your vision or your ability to do math or a chunk of your memory. But you survive.
Right. You're still you, just a diminished version. But if you damage the hypothalamus,
You die.
you die. Or, at the very least, you lose the ability to regulate the basics of life. You can't control your temperature, your water balance, your heart rate. Your body forgets how to be a body. You lose homeostasis.
So let's break down these vegetative functions, because it's not just one thing. It's like a master control panel for the whole ship.
Yeah.
First on the list, cardiovascular regulation.
The hypothalamus is constantly tweaking your heart rate and your blood pressure. If you stimulate the posterior and lateral parts of the hypothalamus, arterial pressure and heart rate go way up. It's the rev up signal. Danger, pump more blood.
And what's the calm down signal?
That's in the preoptic area. Stimulating that area causes a decrease in heart rate and pressure, and these signals get transmitted down through those reticular regions in the pons and medulla we talked about earlier.
So the CEO, the hypothalamus, calls the manager, the reticular formation, who then tells the workers, the heart and blood vessels, what to do.
It's a perfect chain of command.
Okay, next on the control panel, body temperature. This is something we completely take for granted. We stay at, 98.6 degrees Fahrenheit, whether it's snowing outside or we're at the beach in July?
We do, but only because the preoptic area is working around the clock. It contains these incredible heat-sensitive neurons. They are biological thermometers.
And they're measuring the temperature of the blood.
Exactly. If the blood flowing through that area is too hot, these neurons start firing like crazy.
And what's that firing to? What's the response?
It triggers all the cooling protocols. It increases sweating. It dilates the blood vessels in your skin to dump heat into the environment.
And what if the blood is too cold?
They stop firing. And that lack of firing is its own signal. It triggers the heating protocols, shivering to generate heat, constricting blood vessels to conserve it. It's a thermostat. It's literally a biological thermostat.
And if it breaks, you're just at the mercy of the outside world's temperature.
Completely.
Okay, water regulation. This one is huge. The source breaks it down into 2 parts, the conscious feeling of thirst and the unconscious control of excretion.
Thirst and excretion. Let's start with the thirst center. It's located in the lateral hypothalamus. And its job is to sense the electrolyte concentration of your body fluids.
Basically how salty your blood is.
Exactly how salty it is. If you get dehydrated, your blood becomes more concentrated, more salty. And by the process of osmosis, water gets sucked out of the neurons in this thirst center.
Wait, the neurons themselves physically shrink?
They physically shrink. Yes, and that shrinking triggers an intense firing pattern that your conscious mind interprets as one of the most powerful drives we have. I am thirsty. I need water right now.
That is just wild. The feeling of being parched, that desperate urge to drink, is literally a group of shriveled up neurons in my brainstem screaming for help.
That's exactly what it is, but that's only half the story. There's the unconscious side, too. The supraoptic nuclei control excretion.
How does that work?
If your body fluids are too concentrated, those neurons also fire. But they send their signals down a long axon to the posterior pituitary gland.
And the pituitary gland releases a hormone into the blood.
It releases ADH antidiuretic hormone. It's also called vasopressin. This hormone travels through your bloodstream, gets to your kidneys, and gives them a very clear instruction.
Stop releasing water.
Stop releasing water. Reabsorb it back into the body. We are running low. So your urine becomes much more concentrated to save every last drop of water for the body. It's a perfect closed-loop system.
Okay, the next function here feels distinctly mammalian. Uterine contractility and milk ejection.
This is handled by a different set of nuclei, the paraventricular nuclei. Stimulation here causes the secretion of oxytocin.
The famous love hormone.
It has many roles, yes, but in this context, it has two very specific, very powerful mechanical functions. First, it causes the powerful uterine contractions during labor that push the baby out.
Right.
And second, it contracts the tiny ductules in the breasts to actively expel milk.
That's the letdown reflex.
It's the letdown reflex. And just think about the beautiful wiring of that system. A baby suckles. That sensory nerve impulse travels from the nipple all the way to the hypothalamus. The hypothalamus releases oxytocin into the blood. The oxytocin travels to the breast and pushes the milk out for the baby.
It's a direct neurological link between the mother's brain and the infant's immediate survival.
It's an incredible piece of biological engineering.
Speaking of survival, that brings us to the last major vegetative function, gastrointestinal and feeding regulation. And honestly, this section explains so much about the struggles people have with diet and weight.
It really does, because fundamentally this is a battle between two competing centers. First, you have the hunger center, which is in the lateral hypothalamus.
The go signal for eating.
The go signal. If you electrically stimulate this area in an animal, it develops a voracious appetite. It will eat and eat and eat.
And what happens if you destroy it? If you make a lesion there?
The source uses a chilling term. Lethal starvation.
Lethal starvation.
The animal will simply refuse to eat. Even if you pile its favorite food right in front of its nose, it will show no interest. It will starve to death, surrounded by food, because the fundamental drive to eat has been erased.
That's terrifying. It's not that it isn't hungry in the normal sense, it's that the entire concept of eating has been deleted from its brain.
Correct. Now, on the flip side, you have the satiety center.
The stop signal.
The brake pedal, located in the ventromedial nuclei. If you stimulate this area while an animal is eating, it will suddenly stop mid-chew and show complete indifference to the food.
So what happens if you destroy that satiety center? If you cut the brakes?
Then you get voracious eating and extreme obesity. The animal never feels full. It will eat until it is physically, pathologically obese. It just can't stop.
It is just amazing how mechanical this all is. We think of hunger as this complex, nuanced feeling, oh, I'm in the mood for Italian tonight. But at its absolute core, it's just these two centers fighting for dominance. Eat versus stop.
And there's a third player, the mammillary bodies, which control the actual physical reflexes of feeding, like licking your lips and swallowing. So you have the drive, the brake, and the mechanics, all controlled from this tiny 4-gram region of the brain.
So we've got the machine turned on by the brainstem, we've got the chemical mood set, and we've got the body's basic survival regulated by the hypothalamus. But there's another thing that regulates us. Time. The source calls this the master clock.
Ah, yes, circadian rhythms. We all have this internal, roughly 24-hour cycle of sleep and wakefulness. And for a long time, people just assumed this was a passive reaction to the sun coming up and going down.
But it's not. It's generated from inside us.
It's entirely internal. The structure responsible is the suprachiasmatic nucleus, or SCM.
Located right above the optic chiasm, which is where it gets the name suprachiasmatic.
Exactly. And the source mentions that the mechanism here is Nobel Prize-winning science, because this isn't just about neurons firing. This is happening at a genetic molecular level.
It's fascinating. How does a gene tell time?
It's an elegant negative feedback loop. Inside the cells of the SCN, you have a pair of genes called CLOCK and BMAO1.
Okay.
They bind together and they act as transcription factors, meaning they turn on the production of other genes, Specifically, two genes called PER and CIY.
So KLOCK and BML1 turn on the factory to start making PER and CRY proteins.
Right. But here's the clever part. As the PER and CRY proteins start to build up in the cell cytoplasm, they eventually travel back into the nucleus, bind to the CLOC-BML1 complex, and inhibit it.
So they shut down the very factory that made them.
They turn off their own production.
Yeah.
And then, over the next several hours, those PER and CRY proteins naturally decay and disappear.
And once they're gone,
The inhibition is lifted, CLOCK and BML1 become active again, and the whole cycle starts over.
and that entire cycle, make the protein shuts down production, protein decays, start again, takes about 24 hours.
Roughly 24 hours, that is the biological day. It's a tiny molecular hourglass inside each of those neurons, constantly flipping itself over.
But our internal clocks aren't perfect, are they? If you put someone in a deep cave with no light cues, their day might be 24.5 hours or 23.5. We drift out of sync with the world.
We would, so the clock has to be reset every day. That's a process called entrainment.
And that's where our eyes come in.
Yes, through a specific pathway called the retinohypothalamic tract. It carries light signals directly to the SCN.
This isn't from the normal cells we use for vision. They're rods and cones.
No, it's from a special set of ganglion cells in the retina that contain a pigment called melanopsin. They don't see shapes or colors, they just sense the general ambient level of light.
And they send a signal to the SCN that basically says, hey, the sun is up.
Reset the clock now, and that little nudge of light adjusts the timing of that protein cycle. That's how we solve jet lag. We use bright light to force the chemical clock in our brain to align with the sun in our new time zone.
It is wild to think that deep inside our brain, there are these little proteins just building up and breaking down, ticking away the seconds of our life, keeping us tethered to the rotation of the planet.
It grounds all of our complex behavior in a physical, astronomical reality.
Speaking of behavior,
Yeah.
I've talked about the vegetative stuff, the hunger, the thirst, the temperature, but the hypothalamus is also a primary driver of our emotions. The source calls it the emotional driver.
This is where we see the raw primal nature of this structure. It's not just about keeping the body alive. It's about how that body reacts to the world around it.
And the source describes the effects of stimulating different parts of the hypothalamus. This isn't subtle stuff. It's drastic.
It is not subtle at all. If you put an electrode in the lateral hypothalamus and stimulate it at a high level, you don't just get hunger. You get what the text calls overt rage.
Rage. Just pure anger.
Primal fury. The animal will assume a defensive posture, claws extended, hair standing on end piloerection, and it will hiss and spit and attack anything in front of it.
And this is with no actual threat present. You just add a little electricity and this happens.
No threat whatsoever. The feeling of rage is generated entirely internally. The animal doesn't know why it's angry. It just is angry.
And what about the ventromedial nucleus? That was
Yeah.
the satiety center, the stop eating signal.
If you stimulate that area, you get the complete opposite of rage, tranquility. Yes. An animal that is being provoked, even attacked, will just sit there blissfully calm if that nucleus is being stimulated. You can poke it and annoy it, and it has no reaction.
So we literally have a rage button and a tranquility button wired into our brains.
Essentially, yes. And there are others. There's a periventricular zone where stimulation leads to intense fear and punishment reactions. The animal acts like it's being hurt even when it's not.
And sexual drive?
That can be stimulated from the most anterior and posterior portions of the hypothalamus.
So what happens if these areas are broken? We talked about lesions causing starvation or obesity. What's the emotional impact?
It's generally the inverse of stimulation. If you create a lesion in the lateral hypothalamus, the area where stimulation caused rage, the lesion causes extreme passivity.
So the animal loses its drive.
Completely. It won't fight, it won't eat, it just exists. All of his oomph is gone.
And if you lesion the ventromedial nucleus, the tranquility center,
You get a creature that is described as savage and vicious. It displays extreme rage at the slightest provocation. Without that tranquility center to put the brakes on, the rage system runs wild.
this is starting to paint a really clear picture of our personality as this delicate balance between competing centers. If your rage center is naturally a bit more active or your tranquility center is a bit dampened, that fundamentally changes who you are as a person.
It changes your entire affective reactivity to the world. And this leads us to what is maybe the most philosophical part of the source material, the concept of reward and punishment.
The source puts it very bluntly. It says the brain classifies all sensory information as either pleasant which is a reward, or unpleasant, which is a punishment. And this one distinction dictates everything we do.
It's the absolute basis of learning. It's the carrot and the stick, hardwired. We are programmed to gravitate toward things that stimulate the reward centers and to flee from things that stimulate the punishment centers.
Let's look at the anatomy of the stick first. The punishment centers, where are they?
The primary center for punishment is an area of central gray matter that surrounds the aqueduct of Sylvius in the mesencephalon. And it extends up into those periventricular zones of the hypothalamus we just mentioned.
And stimulating this causes that rage pattern we talked about before.
Yes. The hissing, the spitting, the defensive posture.
Yeah.
It's a powerful defense and escape mechanism. And the source notes something really important here. Punishment and fear can, and often do, take precedence over pleasure and reward.
Explain what that means.
Evolutionarily, it's a no-brainer. If you ignore a piece of food, you might get hungry. If you ignore a predator, you get eaten.
So the negative stimulus has to be stronger.
It has to be. The punishment centers are incredibly potent. They can override almost any other drive.
Okay, what about the reward centers? Where's the good vibes button located?
Primarily in the lateral and ventromedial hypothalamus.
Which is ironic, right?
Yeah, because we just said those same areas are involved in rage and satiety. It seems to be a matter of intensity. Mild stimulation of these areas is highly rewarding, but strong stimulation can tip over into these other behaviors like rage. The medial forebrain bundle is also a major pathway for reward signals.
I want to ask how this relates to tranquilizers. Drugs like chlorpromazine are used to treat certain psychotic states. How do they fit into this reward-punishment model?
Well, the theory is that these substances, like chlorpromazine, act by inhibiting both the reward and the punishment centers. They decrease your overall effective reactivity.
So they don't just stop the bad feelings, they actually turn down the volume on everything, good and bad.
Exactly. They dampen the entire limbic system's response to the world. You care less about the punishment, but you also care less about the reward. You become emotionally flattened.
This all brings us to memory. The Force makes a huge point about habituation versus reinforcement, and this directly connects emotion to learning.
This is the brain's fundamental filter for reality. Our senses are bombarded with information every single second. The hum of the lights, the feeling of your chair, the background noises.
If we paid attention to all of it, our brains would just crash.
We couldn't function. So the brain has to decide what's important enough to keep and what to just throw away.
And how does it make that decision?
If a sensory experience causes neither reward nor punishment, if it's emotionally neutral, the cortex learns to ignore it. This is habituation. The response fades away. The source claims we throw away over 99% of all sensory information.
99%.
We simply don't store it.
Yeah.
It doesn't matter. But if a signal causes pleasure or pain, the response is reinforced. The brain puts a tag on it that says, "This is important. Save this."
So we literally only learn what matters to us emotionally.
Correct. Emotion is the chemical tag that tells the memory system, "Save this file." If you don't have an emotional response to something, you are very unlikely to remember it.
And the librarian who does all that saving and tagging, that's the hippocampus.
The hippocampus. Anatomically, it's this elongated structure on the medial part of the temporal lobe, kind of folded inward on itself.
What's its main job in this whole system?
The source describes it as the decision maker. It takes in all this sensory information and determines its importance. It asks the critical survival questions. Is this a threat? Is this food? Is this a potential mate? It directs the brain's attention to what matters.
And the text also mentions it's hyperexcitable, that it's prone to seizures.
Yes, it has a very low seizure threshold. This can lead to what are called psychomotor seizures.
And these are different from the big seizures we normally think of.
Very different. They don't always cause a loss of consciousness. Instead, because the hippocampus is so involved in memory and sensation, a seizure there can cause powerful hallucinations. Olfactory, visual, auditory.
So people will smell things that aren't there.
Yeah.
Or hear voices.
Exactly. It's the hippocampus misfiring and randomly retrieving old memories or creating new sensations from scratch. It's a stark reminder of how fragile our perception of reality really is.
But the most critical role of the hippocampus, the one it's most famous for, is memory consolidation. And the source explains this by looking at what happens when you remove it.
Anterograde amnesia. The source describes patients who had their hippocampi removed bilaterally, that is, on both sides of the brain, usually as a last resort treatment for severe, life-threatening epilepsy.
This is based on the famous case of patient HM, even though the text just describes the general phenomenon. And what happens to these patients is that they keep all their old memories.
Everything stored before the surgery is safe. It's already been written to the hard drive of the cortex. They know who they are. They remember their childhood.
But they cannot form new declarative memories, new memories about facts or events.
That's right. So if you meet one of these patients, you can have a perfectly normal conversation. But if you leave the room and come back 5 minutes later,
They have absolutely no idea who you are. The slate has been wiped completely clean. They are trapped forever in the present moment. Why? What is the hippocampus actually doing in that process that's now missing?
the leading theory is that the hippocampus provides the drive that translates short-term memory into long-term memory. It rehearses the important information over and over, strengthening the synaptic connections until that memory is permanently stored in the cortex.
So without the hippocampus, it's like trying to save a file to a computer, but the save button is broken. The information just sits in the temporary RAM and then vanishes.
It never gets written to the hard drive. That's a perfect analogy.
That is just tragic. But it really drives home the point that memory isn't a passive storage bin. It's an active process, and it's driven by this emotional limbic part of our brain.
It absolutely is.
We have one last major structure to cover here, what the source metaphorically calls the window, the amygdala.
The amygdala. It's a complex of small nuclei located just beneath the cortex in the anterior part of the temporal lobe.
And it has a strange evolutionary history, doesn't it? Didn't it start out as part of the smell system?
It did. Originally, it was an extension of the olfactory system. In lower animals, smell is the primary sense for survival. Does this smell like food? Does this smell like a predator? Does this smell like a mate?
Right.
But in humans and other primates, it's evolved to handle more general behavioral awareness. It helps us navigate our complex social world.
The source lists the effects of stimulating the amygdala, and it's kind of a mixed bag.
It is. You get some involuntary movements like licking, chewing, swallowing. You get visceral changes like spikes or drops in heart rate. But you also get powerful, full-blown emotions: rage, escape, punishment, severe pain, fear.
And the source also notes very distinctly sexual activities, erection, copulatory movements.
Yes, but the most revealing information about the amygdala's true function comes from what happens when it's destroyed. This leads to the famous Kluver-Bucy syndrome.
This was first observed in monkeys, right?
Yes. Researchers removed the anterior temporal poles, a procedure which destroyed the amygdala on both sides. And the change in the monkey's behavior was absolutely shocking. It was like their entire social and survival filter had been removed.
What were the symptoms? What do they do?
There were several key changes. First, they showed extreme fearlessness. A normal monkey is terrified of a snake. These monkeys would approach a snake without any hesitation at all.
So their danger filter was just gone.
Completely gone. Second, extreme curiosity. They would investigate everything, no matter how trivial or dangerous. Third, what the researchers called oral tendencies.
They put everything in their mouths.
Everything. Rocks, feces, nails. Like a baby who hasn't learned yet, trying to figure out if something is food.
In the fourth symptom.
An extreme and indiscriminate sex drive. The source says they would attempt to copulate with immature animals, with animals of other species, and even with inanimate objects.
Wow. So the social appropriateness filter is completely gone too.
Completely. And that's what this syndrome reveals. The amygdala seems to be the behavioral awareness area. It projects the correct behavioral response for any given occasion. It's the part of your brain that tells you, don't eat that, it's a rock. Don't attack that, it's your boss. Don't try to mate with that, it's a chair.
So without the amygdala, the brain is just this bundle of raw drives with no context, no sense of what is appropriate.
Exactly. The amygdala provides the context for our drives. It tells us where and when it is safe and socially acceptable to act on them.
This has been heavy. We've gone all the way from the brainstem's power switch keeping us awake, to the chemical soup that sets our mood, to the hypothalamus regulating our water and our rage, to the hippocampus deciding what we remember, and the amygdala keeping us from eating rocks.
It's an incredibly complex and interconnected web.
So let's try to bring it all together. After this whole deep dive, what's the big takeaway for you?
For me, the synthesis is this. We so often think of things like character or personality as these abstract, almost spiritual concepts. We like to think we are good people because we choose to be good. But this deep dive really shows that our complex personality, everything from our memories and fears to our simple ability to wake up in the morning, is grounded in specific physical hardware.
It's not magic. It's circuitry.
It is circuitry. You feel awake right now because your reticular formation is firing. You feel hungry because your lateral hypothalamus is active. You feel afraid because your amygdala says you should.
And if you tweak that circuitry,
Mm-hmm.
a little lesion here, a bit more dopamine there, the person you are can change completely. The you that you think is so solid and permanent is actually incredibly fragile.
That's the profound and maybe slightly unnerving realization. We are biological machines. Beautiful, wondrously complex machines, but machines nonetheless.
I think I want to leave our listeners with one final thought on that, something to chew on. We talked about the rage button and the tranquility button in the hypothalamus. If a tiny jolt of electricity can turn a peaceful creature into a rage monster, or if a tiny lesion can turn that same rage monster into a completely passive being, where does our character actually end and our biology begin?
That is the ultimate question, isn't it? Are we the sum of our choices, or are we just the sum of our neurochemistry?
Something to think about the next time you get angry. You're not just in a bad mood. Your lateral hypothalamus is actively winning a biological battle against your ventromedial nucleus.
Indeed it is.
That's all the time we have for this deep dive. Thanks for listening to us unpack the machinery of the mind.
It's been a pleasure. Keep those synapses firing.
We'll see you next time.